Now, several of you asked questions about what happens when we were talking about the two-slit experiment, what happens if there is more than one slit? Well, and that's what a diffraction grating is. Basically, you have a bunch of slits, and they're all these little slits, and they're each acting as a source, all right? And they each have, the slit width is still D, but there are a bunch of different ones. The interference pattern is kind of interesting for diffraction grating. If you've got a screen over here, what it's going to look like is that there's, a maximum, and then kind of like it waves around up here, and then there's another big maximum, and it waves around, and there's another big maximum, and so this is called the primary maximum, and then there are also a bunch of little secondary maxes going on in here as there are little alternating light and dark regions here, but it's still fairly dim throughout this whole region before it gets up to the other maximum. So that's called not primary maximum, the principal, I apologize, principal maximum and secondary maxima. Now, these have a lot of slits, on the order of thousands per centimeter, or they can have up to thousands per centimeter. And typically they'll call them lines, because these slits will look like lines on a little thingamajobber. If a thing says they're X lines per centimeter, well, then you just convert centimeters to meters, and then take a reciprocal to find what that separation between each slit is. Okay. D in this case is not the width of the slit, it's the distance between the openings in your grating. We're assuming that this width is very, very small. All right. And just like for our two-slit, these principal maxima, we can do some of the same geometry and trigonometry, but the principal maxima, which are bright fringes, show up where d sine theta is just a multiple of the wavelength in question. Okay. And note that this is related to the wavelength, so that means that if you have white light, it'll spread it all out. Each wavelength will have different places where constructive and destructive interference occur, and so a bright, the principal maximum for blue will be at a different place than principal maximum for green, and so forth. And so you see kind of this rainbow going on when you have a diffraction grating. Using X-rays is of particular importance because is of particular importance in analyzing crystal structures, because the distance involved in natural crystals, these atoms are usually on the order of 10 to the minus 10th meters wide. And for diffraction to be significant, the size of the structure in question has to be on the same order of magnitude as the wavelength, and X-rays have a wavelength on the order of 10 to the minus 10th. So X-rays diffract pretty well through crystal structures. And analyzing how these rays diffract can provide information about the molecule in question. So this is a technique that's used in analysis of molecules and atoms and whatnot. You've probably learned about how the DNA was discovered, and they used a lot of X-ray diffraction. Watson and Crick are credited with discovering the double helix, not how DNA was discovered, but discovering that it had a double helix structure. Rosalind Franklin, who, as I understand it, pretty well got shafted in this whole thing because she was a woman, and didn't wind up getting credit for anything until, recently. But Rosalind Franklin was the one who did the X-ray diffraction. And her data was certainly essential in determining the double helix structure of DNA. Watson and Crick were of course also involved in that, and they did a lot of work as well. But she doesn't quite get the credit that she deserves. Now, diffraction gratings, they do create wide separation between the colors, and there's something called a grating spectroscope that uses this fact to it measures wavelength by how much the grating spreads stuff out, but it's actually really accurate. This grating spectroscope, and gratings are also used in astronomy. A grating spectroscope, is used in astronomy a lot of times to determine what is the main color that a given star or other object is radiating. Diffraction is also why CDs have, if you look at the back of a CD, it appears to have that sort of rainbow color. And that's because when light reflects off the back of a CD, the CD has those lines kind of etched into it, and the gaps between those grooves act as multiple sources of light, just like in a diffraction grating. So that's why you see that sort of rainbow pattern if you look at the back of a CD.